Good day, this is the Professor, CBMD PhD, and today is Tuesday, April 25th, 2023. It is 4:33 and 30 seconds, a.m. Eastern Daylight Time, and today's research, blood cancer. So let's jump to it here. Drug combination restores ability of leading treatment to signal for death of blood cancer cells. Future clinical trials to determine if combination, chloroquine, and venetoclax addresses disease comeback. Date April 24, 2023. NYU, Grossman School of Medicine. Now new research using human tissue samples and mouse models has found that resistance of leukemia cells to a widely prescribed drug called venetoclax occurs because of a rapid increase in the breakdown and turnover of mitochondria, structures inside the cell that help power its functions. In addition to their role in producing energy, mitochondria also tell cells to die under certain adverse conditions. And this process of programmed cell death often goes wrong in cancer. Damaged mitochondria can also undergo a form of cell feeding, termed mitophagy, that prevents them from sending death signals. The studies show that mitophagy helps leukemia cells to evade the killing effects of venetoclax, a drug in a class of medications known as BH3 mimetics. Researchers found that the levels of several genes associated with mitophagy were increased in 20 leukemia patient samples compared with normal controls. The level of these genes was even higher in samples from leukemia patients with drug resistance than in those leukemia patients who were not. Particularly notable was the increased expression of the gene for mitofusin 2, MFN2, which codes for a key protein in the outer mitochondrial membrane. As further experiments using mice in the which bone marrow from acute myeloid leukemia patients was transplanted showed that the drug chloroquine, a known mitophagy inhibitor, restored the ability of venetoclax to kill the cancer cells. Overcoming resistance to BH3 mimetic drugs like venetoclax is a unique clinical significance because these medications are often used for treating people with acute myeloid leukemia. As acute myeloid leukemia is notoriously difficult to treat, with fewer than a third of those affected living longer than 5 years after their diagnosis, so it is important to maximize the impact of existing therapies. The research team plans to design a clinical trial to test whether chloroquine, when used in combination with venetoclax, prevents drug resistance in people with acute myeloid leukemia. And speaking about other study results, the researchers say they not only found that MFN2 was overly active in people with drug-resistant disease, but also that cancer cells exposed similar cell death-inducing components demonstrated a doubling of mitophagy rates. Additional testing in cancer cells engineered to lack MFN2 showed increased sensitivity to drugs similar to venetoclax compared with cells that had functional MFN2. The new study and previous research by the team showing misshapen mitochondria in the drug-resistant leukemia cells confirmed that increased mitophagy was the source of the problem. As acute myeloid leukemia, the most common form of adult leukemia, originates in the bone marrow cells and involves the rapid buildup of abnormal blood cells. The blood cancer results in the deaths of more than 11,500 Americans annually. Current treatments include chemotherapy and a limited number of targeted drug therapies. Bone marrow transplantation has also been used when other options fail. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.